Hello, and welcome to Fun Facts Online. Today, we are skipping the preamble and diving straight into what is arguably the most significant shift in medicine since the discovery of antibiotics. We are talking about a revolution in pharmacology that has moved us from the age of occupancy to the age of degradation. If you follow medical science, you know that for the last century, drug discovery has operated on a very specific paradigm. We call it occupancy-driven pharmacology. Imagine a disease-causing protein as a lock. To stop it, we had to design a drug that acts like a key, or perhaps more accurately, like a piece of gum jammed into that lock. The drug sits in a functional pocket of the protein, blocks its activity, and we hope for the best. This method works for enzymes and receptors, which are what we call the low-hanging fruit of the human body. However, this approach has a massive, fatal flaw. It requires the protein to have a deep, accessible pocket. The problem is that approximately 80% of the human proteome, the entire library of proteins your body produces, does not have these convenient pockets. These include scaffolding proteins, transcription factors, and other flat, smooth structures that pharmaceutical companies have historically labeled as undruggable. For decades, these targets were out of reach. But as we settled into the mid-2020s, that label became obsolete. We have entered the era of targeted protein degradation, or TPD. This is not just a new class of drugs, it is a new physics of drug discovery. Instead of trying to inhibit a protein by sticking to it, TPD technology hijacks the cell's own waste disposal machinery to seek out, tag, and physically destroy the disease-causing protein entirely. It is the difference between handcuffing a criminal and erasing them from existence. While a technology called PROTACs paved the way, the year 2026 has ushered in a new era with the emergence of SupTACs, or supramolecular targeting chimeras. If PROTACs were the invention of the guided missile, SupTACs represent the development of programmable, stealth-capable drone swarms that can strike specific targets in specific tissues at specific times. In this episode, we are going to break down the science, the history, and the explosive future of how we are moving from drugging the undruggable to degrading the indestructible. To understand where we are going with SupTACs, we first need to master the foundation, which begins with the biology of destruction. Every single cell in your body operates a microscopic recycling plant. Proteins are not immortal. They are born, they do their work, and eventually, they become damaged, misfolded, or obsolete. If these old proteins were allowed to pile up, the cell would die. So, to prevent this cellular clutter, the body employs the ubiquitin proteasome system, or UPS. You can think of this system as a municipal waste management crew. It has three main components. First, there is ubiquitin. This is a small regulatory protein that acts as a tag. In the world of cell biology, we call it the kiss of death. Once a protein is tagged with a chain of ubiquitin, its fate is sealed. The second component is the E3 ligase. These are the enzymes responsible for actually attaching that tag. There are over 600 E3 ligases in the human genome, and they act as the gatekeepers. They decide what stays and what goes. Finally, there is the proteasome. This is a massive, barrel-shaped protein complex that acts as the shredder. It recognizes the ubiquitin tag, pulls the protein inside, and shreds it into harmless amino acids, which the cell can then reuse to build new proteins. This system is elegant and efficient, and this is exactly what scientists decided to hack. Enter the PROTAC, or proteolysis targeting chimera. A PROTAC is what chemists call a heterobifunctional molecule. Visually, you can imagine it as a chemical dumbbell. It has three distinct parts. On one end, you have the warhead. This is a ligand that binds to the protein of interest, the disease target. Now here is the critical difference from the old way of making drugs. Because we are not trying to block the protein's function, the warhead does not need to find a special active site. It does not need to jam the lock. It just needs to grab onto the protein anywhere, on any surface. On the other end of the dumbbell is the anchor. This ligand binds to one of those E3 ubiquitin ligases we mentioned, usually one called cereblon or von Hippel-Lindau. Connecting these two ends is the linker, a flexible chemical chain. The mechanism of a PROTAC is an event-driven protocol. It works in 4 steps. First, the ternary complex formation. The PROTAC binds both the target protein and the E3 ligase simultaneously, pulling them into uncomfortable proximity. It forces the executioner to stand next to the victim. Step 2 is ubiquitination. Because they are forced close together, the E3 ligase does its job and transfers ubiquitin molecules onto the target protein. Step 3 is degradation. The tagged protein is recognized by the proteasome and shredded. And step 4 is recycling. This is the most magical part. The PROTAC itself is not destroyed in the shredder. It is released intact to go find another copy of the target protein and repeat the process. This catalytic turnover means that a tiny amount of drug can destroy a massive amount of diseased protein. This is a significant advantage over traditional inhibitors that must remain bound one-to-one to work. As of early 2026, this is no longer just theory. The PROTAC promise has turned into clinical reality. Drugs like vepdegestrant, developed by Arvinas and Pfizer for breast cancer, have shown the ability to degrade the estrogen receptor significantly better than the standard of care. Another drug, bavdegalutamide, targeting the androgen receptor for prostate cancer, proved that TPD works in humans, specifically in patients who had become resistant to all other therapies. We are also seeing degraders for immunological diseases, expanding the horizon far beyond oncology. However, despite their brilliance, first-generation PROTACs face significant hurdles. The first is something known as the hook effect. It is a strange paradox where using too much drug actually stops it from working. At high concentrations, the PROTAC molecules saturate the target and the ligase separately, preventing them from meeting. This results in a bell-shaped dose-response curve, where more drug eventually leads to less effect. The second problem is a lack of tissue specificity. A PROTAC administered systematically will degrade its target in every tissue where the E3 ligase is present. If your target is a cancer driver in the tumor, but essential for life in the heart or liver, a standard PROTAC causes dangerous toxicity. And third, once in the body, a PROTAC is always on. There is no switch to turn it off. This is where the science of 2026 has made its most dramatic leap. In January 2026, a groundbreaking study published in the journal Cell by the team at the Chinese Academy of Sciences unveiled SupTACs. This stands for supramolecular targeting chimeras. This technology solves the two biggest problems in TPD: spatial control, meaning where it works, and temporal control, meaning when it works. So, what is a SupTAC? Unlike a PROTAC, which is a single small molecule, a SupTAC is a modular system that self-assembles into a supramolecular nanoparticle. You need to imagine the PROTAC not as a single dumbbell, but as a swarm of components that come together to form a degradation machine only under specific conditions. These structures use host-guest chemistry, or metal-organic coordination, to assemble multiple warheads and E3 recruiters onto a scaffold. This creates a proximity effect on steroids. Because the nanoparticle brings multiple E3 ligases and multiple target proteins together, it creates a multivalent interaction. This amplifies the degradation signal significantly, allowing for the destruction of proteins that traditional PROTACs struggle to tag. But the true genius of SupTACs is their programmability. Let's talk about the spatial breakthrough. The nanoparticles can be engineered to accumulate in specific organs. In that landmark 2026 study, researchers designed SupTACs that specifically targeted the lungs. They used this to degrade a protein called ACSL4, which is involved in a type of cell death called ferroptosis. They targeted this protein only in the lungs of mice with acute lung injury. The result was fascinating. The lung inflammation was resolved without damaging ACSL4 in the liver or gut, where it is needed for lipid metabolism. This is the holy grail of drug safety. Then there is the temporal control. SupTACs can be designed with caged components. These are inactive precursors that circulate harmlessly until a trigger signal, like a specific small molecule or light, is applied. This allows doctors to switch the degradation on exactly when needed and let it turn off naturally. SupTACs represent the shift from the chemotherapy model, which is like bombing the whole body, to the surgery model, which is molecular precision. By restricting degradation to the disease site, off-target toxicity is virtually eliminated. The modular design also means you can mix and match warheads. If you want to target a different protein, you just swap the warhead module without redesigning the entire nanoparticle. Now, while PROTACs and SupTACs are the stars of the show, the TPD alphabet soup is expanding rapidly. We have RIPTACs, or Regulated Induced Proximity Targeting Chimeras, developed by Hulda Therapeutics. These introduce a hold and kill mechanism. Unlike PROTACs, RIPTACs do not degrade the target protein. Instead, they bind a cancer-specific protein and an essential protein for cell survival, gluing them together. The essential protein is dragged into a useless complex with the cancer protein, preventing it from doing its job. The cancer cell dies because its essential machinery is gummed up. It turns a cancer cell's own overexpression of a protein into a weapon against itself. Then we have LYTACs, or lysosome targeting chimeras. PROTACs work on intracellular proteins via the proteasome. But what about extracellular proteins or membrane receptors? The proteasome is inside the cell and cannot reach them. LYTACs solve this by hijacking the lysosome. They bind an extracellular protein and drag it into the cell via a lysosome targeting receptor, sending it to the acidic lysosome for shredding. And for massive targets, like aggregated proteins in Alzheimer's or even damaged mitochondria, we have AUTACs. These hijack autophagy, the cell's self-eating mechanism, to engulf and digest entire organelles. As we look at the landscape in 2026, the emergence of SupTACs signals that we are moving past the proof of concept phase of targeted protein degradation and into the engineering phase. The concept of the undruggable target is effectively dead. If a protein has a surface, we can stick a ligand to it. If we can stick a ligand to it, we can attach a degradation tag. Artificial intelligence is now playing a massive role here. Designing these massive molecules is difficult, as they violate traditional rules of drug design. AI is being used to predict the perfect linker length and orientation to maximize stability. And finally, the field is moving beyond cancer. 2026 sees TPD moving into neurodegeneration, clearing tau tangles in Alzheimer's, and virology, degrading viral polymerases to stop replication. We are witnessing the greatest paradigm shift in drug discovery since the invention of antibiotics. Traditional drugs were like jamming a wrench into a machine to stop it. PROTACs taught us how to melt the machine down for scrap. Now, SupTACs have given us the ability to melt down only the specific machines causing trouble, in specific rooms, at specific times. The era of targeted protein degradation is not just about destroying proteins. It is about rewriting the rules of life, death, and healing at the molecular level. For disease-causing proteins, there is nowhere left to hide. That wraps up this deep dive into the molecular machinery of the future. Thanks for listening to G Fun Facts Online.